It's my pleasure to welcome Carol from my group, who will talk about her recent publication, which was a difficult one, which was compiling 3 different experiments into one story. But we are happy it went out, and we think it really makes some insights into what's going on with the microbes on the hyphae of arbuscular mycorrhizal fungi. The floor is yours, Carol.
Can you hear me well? Okay. So hello, everyone. Today I will present the work that we published earlier this year in the journal Frontiers in Plant Science. And with this work, we try to answer a simple but important question for soil ecology. That is, can arbuscular mycorrhizal fungal hyphae help microbes to reach nutrient-rich microsites in soil and help the plants acquire more nitrogen? So I would like to briefly introduce you some concepts from our work. So first of all, we will start with arbuscular mycorrhizal fungi. This group of fungi are mutualistic symbiosis with 70% of the plants. And this relationship is very important because they need the plants to survive, because the plant can supply the fungus with carbon, and in return, the fungi will help the plant to acquire nutrients from the soil, such as phosphorus and nitrogen. And another specific thing about this group of fungi is its limited ability to degrade organic compounds. And this is one of the most important parts of the story. But when this fungi is growing through the soil, they can shape a specific microenvironment that we called hyphosphere. And they can shape this microenvironment because they are able to release some exudates in the soil. So they shape the microorganisms that will live close to the hyphae, and also chemical or physical characteristics in the soil in this specific part close to the hyphae. But we need to add another problem here, because this bacteria that will help the fungi, because as I mentioned before, they have this limited ability to degrade organic compounds. So they rely on bacteria to degrade these nutrients, and then they can uptake and send it to the plant. But the bacteria dispersal is very limited in the soil. So again, as this fungal grow through the soil, they can connect distant soil patches and act like roads. So bacteria can use these roads as fungal highways. And we have different types of dispersal, and of course it will depend of each bacteria is there and if it's able to swim or not, etc. So I brought just only one example here, where we have the hyphae of the fungi, and because of the anatomical shape of the hyphae, it can form a thin layer of water above the hyphae, and if the bacteria presents the flagellum, for example, it can swim and reach different soil places. And maybe you are asking yourself why this matters. And this matters because when we think about nitrogen in the soil, this is very challenging. The plant growth, it's totally dependent on the presence of the nitrogen in the soil. And we need this microbial community reach these places where we have the nitrogen, and then they can provide this service that we call mineralization. This is why it's essential. And when it is inefficient or it's not happening, we can have the decrease of the production of the plants. And it's not only important in natural environments, but also when we think about agriculture. And of course, it can lead to an increase in production costs, and we have a lot of environmental problems resulting because of this process. So with this in mind, we have two hypotheses. Higher microbial diversity was expected to promote chitin mineralization and plant nitrogen transfer. And the second one is local recruitment was expected to be more efficient than recruitment depending on microbial migration, which means the microorganisms close to the nutrient will be more efficient than the community that needs to migrate along the hyphae to reach the nutrients. And although each experiment had its own objective, we have some shared characteristics between them. So they have the same microbial inoculum. So we maintain this open pot with a microbial community in our glass house, and we use it in our experiments. They have also the same host plants, the same fungus, the same root-free compartments supplied with 15N chitin and mineral N, and also the sampling and the analysis procedures were the same. But of course, when we analyzed 3 different experiments, it was difficult, but it was good to, it allows us to compare different types of manipulation of the microbial community. So I will try to explain briefly these 3 experiments. In the first one, we had open pots. So there was no protection. It was in the glass house and it was exposed for all microorganisms. And in the second and the third experiment, we have semi-sterile mesocosms. So they were closed in a controlled system. So it was like, I will show you the picture. I think it was easy. So here we have the first experiments, the open pots in the glass house. And then the experiment 2 and 3, they were placed inside these cylinders. So we could control the air entering the system. There was a filter, so we didn't have contamination. And also we use sterile water to put water in the system also. Another important thing about these 3 experiments is the microbial diversity manipulation. So in the first experiment, we didn't have a manipulation of the diversity because it was open. It was receiving microbes from everywhere, but also we prepare filtrates of soil and we inoculated these filtrates of the microbial community. It was not just bacteria. This microbial community in the hyphosphere and in the compartments. But when we started the experiment 2 and experiment 3, we started also to manipulate the community. So we prepared soil filtrate again with the same community. And you can see here, we use the technique that we call size fractionation. So we start with 1 millimeter and then we decrease the size of the mesh and we filtrate this community to try to reduce the diversity. And in experiment 2, we added these different filtrates in the hyphosphere, but we have another characteristics here that is microorganisms in the root-free compartment. So here we used also the microorganisms in the root-free compartment, but only the filtrate prepared with the 2 micrometers mesh. And in the third experiment, we also had the microbial filtrates, but here we didn't apply any microorganisms in the root-free compartments. So to reach the nutrient, the microorganisms need to to use the fungal highway to get there. So what did we find? We first analyzed the 15N transfer to plant, and here in the experiment 1 and in experiment 2, we had experiment 2, and almost all treatments in experiment 2, we had the highest 15N transfer to the plant compared to experiment 3. But when we had the most diverse field traits, we start to see again some activities of this community, and we have a slight increase in the 15N transfer to the plant. But it's also important to note here that we almost didn't see the effect of the effect of the different treatments or the effect of the different diversity of the microorganisms. So we first thought, okay, so maybe in experiment 3, the fungus was not able, he failed to access the soil-free compartments, and this is why we didn't have the 15N transfer to the plants. But it didn't happen. We have the colonization. So in experiment 2, in 1 here, we can see a pattern. So the fungus was able to colonize the chitin compartment and the mineral N compartment. But in experiment 3, we also have it, but he colonized more successfully the mineral compartment. And it was probably because this type of nutrient the fungus cannot take without the mineralization. And looking for the abundance of the bacteria, in experiment 1 and experiment 2, we had the highest colonization again of the prokaryotes. And it's very interesting, but in the experiment 2, they really were abundant in the chitin compartment. But when we looked for experiment 3, it didn't happen. So here, comparing with the hyphosphere, in almost all cases, hyphosphere presented the highest abundance of prokaryotes, so they didn't move. But again, when we look for the most diverse treatment here, we start again to see some organisms that can reach the chitin compartment. And probably it happens because when we filtered the microbial community, we didn't filter only the bacteria, but also the whole microorganisms in the soil. And here we probably reintroduced in the system some motile bacteria that was not present in the other treatments. We also had the presence of streptomyces that present the filamentous growth. So it's slow, but he can stay there along the time. He can arrive in the compartment. Or also we have some microorganisms that can use the hitchhiking, we call, and which means that they can use other microorganisms to have a right and arrive in the root-free compartment, for example, protease or other motile bacteria. And finally, when we checked the diversity of this community, we can see that in experiment 1, comparing to the hyphosphere, we have the highest diversity in both compartments, mineral and chitin compartments. But in experiment 2, we saw a compartment effect. So here, the chitin compartment presented the smallest diversity in this community. In experiment 3, we couldn't see a lot of difference between the hyphosphere and the root-free compartments. But again, when we start to introduce more higher diversity of microorganisms here, we start to see some difference in experiment 3. So coming back for our hypothesis, the first one was not supported because we saw just a few differences between the treatments when involved the reducing the diversity of the microorganisms with the technique, the soil filtration. But the second one was supported because we saw that when we put the microorganisms close to the nutrients, they were more effective to mineralize the nitrogen and transfer to the plant. And it was the opposite when we need to, the microorganisms need to migrate in the fungal hyphae. So the main conclusion here is that fungal hyphae is important, but we have more factors affecting the 15 and transfer to the plants. And in future studies, we need to focus on exudates and its effects on recruiting bacteria, or how it can changes the presence of the bacteria in the hyphosphere, the motility, because the bacteria is always very different from each other. So which types of motility we can have in the soil and in the fungal hyphae, and also the resource sensing, because it's important, and we exactly don't know yet how the microbes needs to be stimulated by the presence of the resource to start to use the hyphae to migrate. Let's do that. Thank you.